can be studied by the pedigree analysis, but not all the time, because now we know that there is a complex genetics, there is even de novo and other mutations that we cannot actually assess through the pedigree analysis, but still. So when we look at the human cell, so each human cell have 46 chromosomes. We have 23 pair of chromosome, and then of course like half is coming from father, half is coming from mother, and then we have 22 pair of autosomes, and autosomes are the one that don't code for the gender, but then the chromosome that code for the gender are sex chromosome, and there are a pair of sex chromosome, XX in case of females and XY in case of males. So what these chromosomes are made of? So chromosomes are made of DNA, and DNA, I'm sure you all know, it's deoxyribonucleic acid, and there are 4 nitrogenous bases. Those are the building blocks of DNA. So all the information that is coded is in the DNA in the form of genes. So, and of course, the genes are transcribed into RNA, and then of course later to the protein and perform the function. And we are going to do, talk about this in detail. So if we look, we know that now there are 46 chromosomes in human genome, and we also know that if we look deeply, that A, T, C, and G sequences in the in one cell, so it's around 3 billion base pair or 3.4 billion base pair according to some estimate in those 23 pair of chromosomes. And these 3.4 or 3 billion base pair, they encode roughly around 25,000 genes. And how they perform the function. So first we have the DNA, and DNA is transcribed into RNA, and RNA is translated later into protein. So this is like the whole central dogma. We have the DNA, it goes through the replication, and then of course it goes through the transcription. And in transcription we have RNA, and then we know that in gene we have exon and intron, the coding and non-coding portion. So the non-coding will be removed, and later we'll have only the coding part, and then will be translated into the protein. So if you look on this 3.3 billion base pair, it's literally like if each cell have a book, or our genome is a book. So these are, and this book have 3 billion time in duplicates, because of course if you further expand, now we are talking about the base pair, but if you talk about the single base, it's around 6 billion base.
Recording in progress.
Like some information in the book or the chapter, for example, the references and these kind of information, maybe it will not be as harmful as if it's in the actually some word that makes sense. And because of this small change, if it's not able to make sense anymore, then of course this can lead to the genetic disease. So yeah. It can be a condition that can present at the birth. And of course, there are diseases, and the onset of disease can vary because of course, the information is in your DNA, but then we all know that there are some diseases that are late onset. For example, the muscular dystrophies, they have a particular age of onset. And for instance, if we go further, we know that the schizophrenia and most of the psychiatric illness, they have a particular age of onset. So most of these genetic disorders are quite rare, and like one person in several thousand or millions is affected, but still these are quite common. Like now we know there are more than 7,000 conditions. Those are rare genetic conditions. And so genetic disorder may be heritable, and now that is, they pass from parents to the offspring, but now we know with this somatic mutations, they are not heritable. So what is difference? For example, this is a normal gene and it encode a normal protein, and then we have mutated gene. There is something wrong here, and that's why it will lead to the abnormal protein and sometimes no protein. So in both cases, it can affect the body function differently. So now we are going to talk about the inheritance pattern and these things. But before going into the detail, there is a person whose contribution to the human genetics cannot be replaced, and he's called father of genetics. And even though it's we have a lot of advancement in genetics, and especially the human genetics, but of course it started with the Mendel's experiment on the pea plant, and he actually came up with the law of segregation. At that time, he didn't know about the genes and all these nucleotide bases and all these things, but the law that he proposed are still relevant. So one is the law of segregation. It's mean the one allele is coming from father and other allele is coming from mother, and they segregate in a different gametes. And then the law of independent assortment is, for example, you have that time he called characters because his work was on pea plants. So for example, there is a gene that is encoding one character and the other gene is encoding another character. So they will assort independently into the gametes. So they are not, although we have the linkage and that is opposite to this law, but still it's relevant. And so some characters are dominant and some characters are recessive. So that we are going to talk. There are a few terms. So Mendelian factors, that time he called factors, but now we know that we call them gene, and the allele is an alternate form of the gene. Then we have individual who have two identical alleles, they call homozygous. And then we have, if there is different alleles, we call it heterozygous. And then genotype is the genetic information, or so it's called the genetic composition is called genotype. And because of that composition, the phenotype or the physical appearance or the character that we see, we call it phenotype. So these are the classical Mendelian terms. So now we have mode of inheritance based on the Mendelian pattern of inheritance. And that is quite relevant, and we still go through all these patterns of inheritance, and they are important, especially for our population, for Pakistani population. And I'm going into detail later, like why our population have a different genetic structure than the rest of the world population. So there are certain mode of inheritance. It's autosomal recessive, dominant, X-linked recessive, and X-linked dominant. So, and how we actually go through this pattern of inheritance, we make the pedigrees, we make the family tree, or like the history of the family with the disease and the parents and the other relationship in the family, in the pedigree. So these are some classical symbols that we use. For example, this is male, female, gender unknown, and if it's affected, so it's mostly filled. And heterozygous, we can actually, if the patient have two conditions, so you can give it a different color, or if you have four, so you can even make it further into 4 colors. So carrier is with the dot, and these are the multiple individual. If we don't know the gender, then this is a sign. If gender is known and male or female, this is a sign. So the proband have an arrow at the end, and then if it's disease, so this is the symbol. Then we have some further, for example, if there are spontaneous miscarriages, if there is termination of pedigree, affected and unaffected both, monozygotic, dizygotic, or the twins where we don't know. And then no offspring, if we have infertility, we know that now a lot of infertility have the genetic cause. This line show the marriage, and then if the marriage is between the related couple, for example, if it's between the cousins, so it have the 2 lines. And then this is the separation, and then of course this is adoption at the end. So these all are the classical pedigree and symbols. On the basis of this, we make the pedigree. And if you are looking at the pedigree and you see any of these signs, you will clearly see that this is showing. And especially this double line in the marriages, it's something that is very common in our population, because almost 70% of our population have the cousin marriages. So why we draw the pedigree? So it actually have multiple function. So the first step is we can see the risk assessment. We can see if this disease is recessive, so how this is going to go further in the next generations. And of course, it's another way to collect the family history. And now we know that there are disorders, those are a little bit different, but still encoded by the same genes. And there are some other environmental factors, so we can actually assess those. And of course, it's also, all the information, and later we go through the carrier screening and all these things, so it make easy for us to make the pedigree. And even once we make the pedigree, and when we are going for the analysis or the identification, we can actually get an idea, like what is the possible pattern of inheritance and what kind of variant or we are expecting. So these are quite helpful. So the first is autosomal recessive, and this is so in recessive and dominant, there are 2 things. Recessive main generation skip neosis. So there is no, so in recessive, we have the skipping of generation, but in dominant, there is no skipping of generation. So that is recessive and dominant thing. And then the autosomal is something that is the gene, because of the gene on the autosome. So of course, it appears in the both sexes equally, and the both gender, either male or female, have the equal chance to be affected. And mostly, since there is the parents are carrier, so it's shows that this is most common in the cousin marriages, and the parents are carrier. So if the both parents are heterozygous, so there are chances, according to Mendelian pattern, that there are 25% chances in each pregnancy of having an affected child. And since it's a 25% chance in each pregnancy, so sometimes it's very difficult to tell people, like when you are doing some genetic counseling and telling people that, oh, you have the risk 25%, it doesn't mean that this is just like 4 out of 1. So because we see the pedigrees and families where they have affected child in each pregnancy, because there is 25% chances. So if we look, there are 7,000 rare conditions, and mostly the autosomal recessive condition, but there are few very common examples like cystic fibrosis, sickle cell anemia, beta thalassemia. And beta thalassemia is again one of the most common genetic disorder in Pakistan, though it's a very rare one, but in Pakistan it's quite common. And now it's so common that it's not because of the cousin marriages anymore, because the population frequency is quite a lot. So if we looked at these, both parents are carrier. So this dark blue at the bottom is affected. So they have one allele for affected or the disease copy. And then, of course, what could be the outcome after each pregnancy? So it will be one unaffected child having both normal allele. And then there are 50% chances that there will be a carrier like the parent. And then 25% chances, because if the child inherit both disease copies, so it is going to be affected. So this is a classical recessive pattern. Then we have dominant, and as I already mentioned that the dominant is something that doesn't skip the generation, and since it's autosomal, so it will appear in both sexes and both genders. So so affected child usually have affected parent, and so in dominant, basically one disease copy is sufficient to cause the disease. So it is appear in the heterozygous condition as well. So there are many conditions, autosomal dominant condition we see. And now there is something very interesting, that the same condition can be caused by dominant and the recessive as well, depending on how crucial that change is, how important that change is in your DNA. So for example, in the same gene, if there is a mutation and it's a heterozygous, sometimes it does not cause the disease. But on the other hand, if it is only causing disease in the recessive form, if the both allele have the disease mutant copy. But even in the same gene, there are some mutation that are like even in heterozygous condition are sufficient to cause the disease. So now in one disease, it can be inherited as autosomal recessive as well as autosomal dominant function. So here we have affected parents, so it have the 50% chances to have the affected child, both sexes again, and then it have 50% chances to have a normal individual because one of the parent is normal. And then we have X-linked recessive. Again, it affects more males. And this is a condition in which there is a lot of male lethal phenotypes, because since we all know that male have one X chromosome, so sometime if there is mutation in X chromosome and it inherit from the mother, so it have the disease. And if the mutation is very important or the gene is very important, so most of the time males doesn't survive. So all the male fetus get during the pregnancy get terminated and lost embryonically because the mutation are embryonic lethal. But it doesn't affect the females mostly, because it have the sons are born to an unaffected mom, because again one copy is sufficient to cause, in male if we have one X, so and it's mean that there is no protein made by that X chromosome. But in female we know that there are two X, so females are carrier and males are affected. So this is like a typical X-linked recessive. A lot of disorders are X-linked recessive again. So we have here some classical example of Duchenne muscular dystrophy that we know that mostly if the mother is affected, there are chances to have 50% of the son affected in each pregnancy. So here we have the female that is have a carrier, and it's mean that it is going to pass through the 50% child. And then if the father is unaffected, and it's mean that and if the mother is affected, so it can inherit that affected allele to the female, and female can be only carrier, like in this case we can see, but the male will be affected. So, and then in last we have the X-linked dominant. So in X-linked dominant, the females and males both are affected. There is no skipping of generation. And when the mother is affected, there is two conditions. So when mother is affected, 50% of her son will be affected. And when the father is affected, all of her daughter will be affected, because father have only one X chromosome. And if that father is affected and he's passing that affected allele to his daughters, because he have only one X, so he doesn't have any choice to give a normal allele. So all of the daughters are going to be affected. So this is the classical pattern. Again, a lot of autosomal dominant condition, Rett syndrome, incognitia pigmenti. And then again, for example, in this gene, it have male lethal phenotype and female are mostly affected. So if the father is affected, so all the daughters are going to be affected. And if mother is normal, so all the son will be normal. But if the mother is affected, so there are 50% chances to have an affected child, either male or female, both sexes. But then we have some other pattern. For example, we know that we have the mitochondrial genome beside our nuclear genome, and that have a pattern that is, so in mitochondrial inheritance, the inheritance is from the mother. So for example, if mother is unaffected and father is affected, so all the child are going to be affected. But if the mother is affected and the father is unaffected, then all the child are going to be affected. But of course, where the phenotype is, this can vary, because of course the degree of mitochondria or the amount of mitochondria they get from mother, so it is going to affect their severity of the disease. So now I would like you to look at this pedigree and tell me if what kind of pattern of inheritance you think that this have. If anyone can tell me. I mean, for example, there is no skipping of generation, then what you think?
Autosomal dominant.
Yeah. So it seems like autosomal dominant because both sexes are affected. There's no skipping of generation, and affected individual have one affected parent. Okay. Now another one we have. What you think about this one?
Mitochondrial inheritance. But
if it's mitochondrial inheritance, then where mother is affected?
X-linked recessive.
Yes. Yeah. Because it's only males are affected, because we have studied that when the females are carrier, so they can actually, all of their sons are going to be, or the 50% of their sons are going to be affected. For example, here we have a normal son as well, but then we have the son with the disease. So it's males are affected, so mother are possibly unaffected carriers. So affected father doesn't have affected son. So otherwise, it can be Y-linked, but of course there are unaffected sons, so it cannot be Y-linked. So this is the pattern. So what kind of information we need to make a pedigree? So for example, we see a case that Ali is a 3-year-old male born to a non-consanguineous parent. We already know that the parents are not related. And you have like delayed walking, muscle weakness, hypotonia, EMG findings have increased CKP levels. So this is the clinical picture of the patient. But then we also need family history. If there is someone else have a disease similar to Ali, or what is the status of the, the immediate family. So how many siblings he have, how old they are. And if, like, for example, sometime if there is a disease onset, at 8 years, for example, it's a muscular dystrophy and it start appearing this disease symptom at 8 years, and the child have a younger sibling, for example, is 2-year or 3-year-old. So it doesn't mean that those are, and for now, if they are like 3-year or 2-year-old and they don't have any symptom, so it doesn't mean that they are normal, because there might be chances that they also have the same genotype, but that genotype will actually appear later in when the age of onset will arrive. So this is the whole information of the individual's family, how many siblings the parents have, and if any of their sibling child have any disease similar. Because, for example, if there is a dominant and it's not coming from family history or the like consanguineous marriage or cousin marriage, so it might be because if the mother have a son that have disease and mother's sister have a son that have a disease, so we can actually think about some X-linked inheritance, X-linked recessive, and these kind of inheritance. So if we have to make, since we don't have much time, it would have been great if we could make like some pedigrees at least. But it's like, if we have to make the pedigree, because there was some different phenotype we have put in this fashion, and we know that this is a line show the marriage, and this is the child, and then if there is a disease, so this is generation like this is first generation, second generation, and third generation. And we have Ali here, so this is a proband, so we have a star here, arrow here, so it's not the full arrow, but of course it needed to be full. And then we have similarly another case where Sana is a 4-year-old girl referred to the genetic clinic, and she have these clinical features, and she is born to consanguineous parents, Sana's parents. And then when it comes to cousin marriages or consanguineous parents, so we all need to ask again when we make the pedigree, so we actually trace like how they are related, whether they are first cousin, where they are second cousin, third cousin, whether there is a generation gap or these kind of things. So she have a younger brother, is a 3-year-old, so parents are expecting again, and a mother is 8th week of gestation, and Sana mother have no brother, no sister. Basically, we don't know like whether the disease is, which kind of a pattern the disease have, unless we don't get the genetic information. So yeah, this kind of pedigree will make this is a cousin. Okay, the pedigree that I made in this software, so it somehow it's kind of a free software, and I made some pedigrees for this one, so it doesn't show the whole kind of black filled symbol. So but you have to think that this is a filled one, because of course it's a patient, and then the mother have this, so it basically cover most of your pedigree symbols. And then there are online tools, for example, this Progeny Genetics is one, there is Cyrillic, there are some other tools where you can actually make the pedigrees, and Cyrillic is something that is, I think, so paid, but it's a very good one. So so what are the end goal, why we make the pedigree? So of course it end the diagnostic, and then it's also for the medical diagnosis in terms of possible treatment. And now while making pedigree, we can actually trace like the risk. We can do the risk assessment. For example, we have if we make the pedigree and we know the pattern and we diagnose the disease that we are going to read later, like how we do that. And once we have the genotype, so we can actually go back to the family and ask about tell them about the risk assessment, and we can do the carrier screening. And of course, we can tell about how how many chances they have to have similar condition. For example, if you have a family with thalassemia and you know the mutation and you know the carrier in the sibling of the parents, so you can definitely tell about the risk of or the reoccurrence of the disease. So before we move to the genetic disorder, do you have any question? Because we have some time.
मैम प्लीज जरा माइटोकॉन्ड्रियल इनहेरिटेंस की कुछ एग्जांपल्स अगर क्लिनिकली आप एक्सप्लेन कर दें.
Mitochondrial inheritance, I mean, I don't specifically remember now any clinical like, you know, the disease name, but definitely this is one area that is now, because of course if you look on the whole gene and genome and you don't find anything, so then you can actually go to the mitochondrial one. And mitochondria ka thoda sa ye hai ki, because of course we can confuse it with the complex disorder or with the oligogenic inheritance, because in that one we have this variety, like a variable phenotype aati hai. But sorry, I don't remember any clinical disease right now with the mitochondria.
It's good.
Okay, any other question?
Ma'am, are neural
Yes.
are neural tube defects genetic, or matlab, are they carried through genetic information?
Yes, there is a lot of neural tube defects. Those are genetic, and one thing that is actually in the neural tube defects is because if the patient have a neural tube defect, and because we know that in early embryonic development there is the genes, those are involved in that time, for example in neural tube, or for example jo EMT transition may involve hai, so they are very, very important. So even in there is a genetic change, like mutation, so that can lead to mostly the lethal phenotype. So there is a huge spectrum of diseases jo ke mostly early embryonic development में involve हैं जो gene, उसकी वजह से जो mutation उन genes में होती हैं, तो and because of those mutation mostly it fetus survive नहीं करते, and we see in the clinic that there is a recurrent pregnancy losses, and even it's, since we know that this is very early in development, तो हमारे पास कोई as such fetal information होती भी नहीं है. So and now recently in the recent year there is a lot of research coming up where we know that yes, all are these have the genetic cause, and not only कि वो survive नहीं करते, even if they are they survive, so they have like early neonatal deaths वगैरह जो होती हैं, because of course mostly they are not compatible with life, but yes. And then there are some who are like, for example, we have this one gene ZEP2, उसमें mutation, एक even small change heterozygous change की वजह से you have epilepsy and a lot of other associated phenotype for it's called Mowat-Wilson syndrome. That's also something that is very early, जो जब EMT transition आपकी हो रही होती है. तो I mean throughout the process of development, जो gene involve हैं, अगर उसमें mutation आएगी तो definitely it's going to cause a disease. ठीक है?
If a couple have constant children, like one after another, and they are having cerebral palsy, so is this a neurogenic defect or what is this?
So previously it was thought कि cerebral palsy जो है उसका कोई genetic इतना link नहीं होता कि, you know, recently I studied this paper where they have actually looked on 250 patients with cerebral palsy and they found the genetic cause. So now अगर उसकी reoccurrence हो रही है, for example, the way you mentioned that if they have like 2 child with a similar disease, and even certainly if there is one child, so genetic cause तो हो सकता है अगर आपको कोई और उसकी explanation समझ नहीं आ रही. So yes, now we know that cerebral palsy have a genetic cause. So it was a paper, I think, so it published in Nature. So they have 250 patients and they found some rare mutation in cerebral palsy patient. So yes, it has genetic cause. Any more question or should we move?
Yes, ma'am, we can move.
Okay, perfect. So now we have studied those pattern of inheritance or genome and DNA and everything. But what are the genetic disorders and what level these genetic disorders occur? So you are all familiar with this thing. It's human cell, and we know that in nucleus we have 46 chromosomes, जो कि हमने already we have looked at. And then if there is, and these chromosomes have genes, so each chromosome have different genes. And then if there is the defect in the chromosome, either it's a whole chromosome or a part of chromosome, then it is going to call chromosomal disorder. But if there is a mutation, if it's a change, either a small change or a big change, what that changes we are going to talk later in this presentation. So that is called Mendelian or rare disease. For example, microcephaly, thalassemia, deafness. But if there are small changes in multiple genes, so for example, in different genes, then it can cause a complex disorder that we call also, and not only these changes, also the environmental factors, those are involved. So they are going to cause either complex disorder or multifactorial disorder. So for example, diabetes, cardiovascular diseases, and other neuropsychiatric conditions. So these are so basically this is something that we are going to talk in the coming slide. So this is probably, if you have any question on this slide, otherwise we can move further, because this is the main 3 types of genetic disorder. And then of course in each genetic disorder बहुत सारी different types के उसके हो सकते हैं ना. For example, in chromosomal we know that there could be complete deletion or addition of a new chromosome, or a small part is deleted, and इसी तरह से. So we call chromosomal and Mendelian disorder, or the monogenic disorder, rare disorder, and complex को हम complex ही कहते हैं. So then when it comes to chromosomal disorders, so we are going to talk about these all disorders one by one. So the chromosomal disorder is the change in number and the structure of the chromosome. So I will know that if the whole chromosome number is changed, for example, in Down syndrome, so it is going to affect the phenotype differently, and it is probably going to be huge. It's not only, for example, in Down syndrome, we have, it's a syndromic condition. It have defect in intellectual disability, it have heart defects, it also have like a lot of other craniofacial features and other like mostly neurocrystopathies. And of course it predisposes the patient for early Alzheimer and some early cancers. So it's a wider phenotype, because the whole set of chromosome पे जितने एक gene थे वो extra आ गए हैं, तो addition or deletion both cause the disease. So for example, this is our normal set of chromosome. So it's just, again, I will give example of a library where we have books, where we have a complete encyclopedia set of human genome. It's male and it's female and the male. So what if we add another book to the shelf, and if we remove a book or like a part of a book, then of course, like for example, this was previously this book 5, or the chromosome 5 was in 2 copies, but now we know that at least the half part of the book is deleted. So this is going to cause the disease. So this is numerical. And then we have like the whole part of the book is not deleted, but uska, the whole chapter or the whole book is not removed, but few pages are either deleted or added. For example, in this case, the page 41 is deleted, 40 is deleted, and in this one, we know that there are certain pages from 31 to 40, for example, they all are deleted. And similarly, in this one, we have there are multiple copies of 35 pages. So it's if you have written a thesis or a report, and when we have to submit multiple copies to the library, and sometimes when we rearrange and we put those pages together, sometimes we skip one page or we add an additional page. So this is similarly what happened with the chromosomal disorders. So either complete absence or like addition or deletion. And so there are different types of these addition and deletion. So one is that, for example, this is the gene on the chromosome, and if there is a deletion of B, so it will be the B, the whole part will be missing. And then if it's a duplication of B, so then again we have an additional gene, and then there is inversion. So instead of B at the right arm, now it's at the left side, and of course the position is changed. And similarly, if it's like the whole information is translocated, it is present now elsewhere, not in the original form. So these all are common chromosomal aberrations or the changes. So we are going to talk about some classical examples of the chromosomal disorder, and then we'll move to the further. So the Down syndrome is quite common. It have 1 in 750 child. It have a lot of, it have a hypocellular brain. It's mean in the brain, because we did some work on Down syndrome as well, and we looked on the brain, and there was less mature neuronal population in the Down syndrome brain as compared to the wild type or the healthy brain. But instead of that, the neuron was less, but then there was another population that was immature neuronal progenitor, and that was quite high. So of course there is जो एक balance होता है from immature to mature, that was disturbed. And of course it also have, like as I already mentioned, the craniofacial changes, and it have some digestive, some heart issues and all these things, and it is an extra whole chromosome 21 is added. Similarly, we have Edwards syndrome. It's also quite common. It is 1 in 5,000 live births. And it's a very severe one, because probably the gene on 18 chromosome are involved in some crucial functions as compared to the gene in chromosome 21, because the 18 probably have role in early development. So that's why most of the child with this extra copy of chromosome 18, they die before birth. They have a lot of functional or multi system abnormalities, like kidney malformation. They have heart defects, intestine defects. They have developmental delay, growth deficiency, feeding difficulty, and breathing difficulties. So of course, very, very crucial function. And if there's an additional copy, so so they are not going to survive mostly. Then we have Lattice syndrome. So it's a little bit less common than the other two, and it's in this one we have trisomy 13. So and again, similarly to the Edwards syndrome, it's also very crucial, because of course it doesn't survive, and few than 5% survive first 3 years of the age. So again, very lethal phenotype, and it also have like a lot of multi-system abnormalities. So these are all that we can actually further going down कि हम इनको identify कैसे करते हैं. And then we have some where we have gender abnormalities in the sex chromosome. For example, we have Turner syndrome, Klinefelter syndrome, triple X syndrome, and we have double Y syndrome or double Y male. So these are all the changes that are either a whole chromosome is added or deleted. But beside this, we have a huge thousands of other disorders where a part of chromosome is deleted, and that part can be bigger, that part can be smaller. For example, this chromosome 5p deletion. So that's also quite common, 1 in 20,000 to 50,000 births. And they have like cat-like cry. That's why that's where the syndrome name came from. Again, small head, round face, typical feature appearance, heart defect, muscular and skeletal problem, hearing, sight problem, walking, talking issues, behavioral issues, and mental retardation. So if you look at the phenotypic spectrum, so we see that agar ek pura whole gene is, a whole chromosome is deleted, and we know that chromosome, for example, chromosome 21 have a couple of hundred genes. I'm forgetting exactly, I think so, whether it was 200 or something genes. And similarly, if that is additional copies have been added, so uska ek alag spectrum hoga. And similarly, if the part of chromosome 5, a P arm is deleted and it again have a huge spectrum, and that's different than the rare Mendelian, the other Mendelian or monogenic disorder, because in that one we have a one gene mutation in a part of one gene only. But in this one we can have the deletion of that span 30 genes. We have deletion, we can have a deletion that is 2 gene, 3 gene, depending on the number of gene, there will be severity of the phenotype. Again, 22q deletion is very common. It's actually associated with a lot of syndromic condition. Again, if we look at the deletion, so for example, this is the one deletion and the other person have a deletion that overlaps to this one. And it's mean like this part where these two deletions are overlapped. So they have shared phenotype, but then the part that is here or here, it's a different phenotype. So yeah, so they also have a huge spectrum of the phenotype. And there is a very common 22 chromosome deletion in neuropsychiatric disorders, especially with the schizophrenia. And do you have any question till here?
No,
if
you're
not-
going ahead.
Okay, I just want to make sure that you are with me, and if there is any question, so please raise your hand or ask. So then we have monogenic disorder, the classical Mendelian pattern of inheritance. Because of mutation in one gene, we have the disease. So the monogenic disorder, also called monogenic trait, or called by variation in single gene. So, and there are approximately 7,000 rare diseases or the monogenic diseases. And now, yesterday I was reading a paper, and it'll have a huge impact, like 50% of all disorders are, the economic burden is on the rare diseases because of the rare diseases. So even if there is a change in single base, imagine out of those 6 billion nucleotide, if the one is changed, so it can lead to a disease that can be lethal depending on, or it can have a phenotype, or it can have a lifelong condition. And that's what monogenic disorders are. So this is a Pakistani situation, and a lot of these monogenic disorders, especially the X-linked recessive disorder, they come from consanguineous marriages or the cousin marriages. So if we look at the world map, this is down is showing the percentage of consanguinity or the the cousin marriages in the world. And if you can see my cursor, it's like the Pakistani is here. So it's among the country in the highest, with the highest consanguineous marriages. And that's why Pakistan have one of the highest rate of these rare genetic condition in the world. So if we even look at, so Pakistan have autosomal recessive genetic condition 8 times higher in the cousin marriages. And if we look on the inbred, the outbred population, so it have like 10 times more than European population that is outbred. And so even if we look at our other Muslim countries who practice the cousin marriages, Pakistan is still among the top, like with the 70% consanguineous marriages. And especially in Khyber Pakhtunkhwa, we have a lot. And of course, the trend is different between the cities and between the, the villages or the other areas, but it's still like around 70% reported consanguinity we have. So what this consanguinity does, and why we always talk about that, because of cousin marriages, we have high risk, we have a lot of genetic diseases. So it's, for example, if there is a mutation in ancestors, in the forefathers, and that mutation travel from one generation to the other generation, and and एक mutation हो गई कहीं ancestor में, and then it's just traveling. So, and if we have the custom of cousin marriages, so there will be chances to have more carrier, and then within the family. And if there is a marriage between two carrier, the typical autosomal recessive inheritance that we have seen, so this is like consanguineous marriages, and then they are going to have affected child. So that's why we have one of the highest risk of genetic diseases in our country. Yeah. For example, why I put the specific disorder, microcephaly. I want to ask a question to you guys first. What you think that microcephaly is? Is it a genetic condition, or it can be because of something else? Because, for example, हमारे जो पाकिस्तान में हम इनको शायद औले के चूहे कहते हैं. So and there are a lot of myths that they put some iron skeleton on the skull, and that's why they have this condition. So tell me, what is your perception about primary microcephaly?
Genetic.
Who think it's not genetic and who think it can be both?
Genetic.
Ji?
No, not genetic.
Okay. Is there anyone who thinks that it's not genetic? So basically,
Ma'am, is this genetic?
yes, it is genetic. So basically, primary microcephaly is classical genetics, Mendelian or monogenic genetic trait. There is nothing coming from putting something on the top of the head or this kind of a thing. So that's all जो है ना वो fake stories हैं. So basically, and this is something that happened very early in development, like, you know, during the development. So of the cerebral cortex, if there is a mutation in a gene, okay, they are involved in any of this, you know, either proliferation or any other process during the development that can lead to microcephaly. And why I put microcephaly here, because we have a huge burden. We have one of the highest incidence of microcephaly is in Pakistan. And in fact, there are 25 genes reported in the world, and most are from Pakistani population. So I mean, again, you can see like out of 25, I think so even the first gene that was reported for microcephaly was from Pakistani population. There's a lot that is coming from Pakistani population. So yes. Okay, now we comes to the mutation. So we have checked that chromosomal mein to either it's the whole chromosome is added or deleted, or a part of chromosome spanning different genes is deleted or added. But what kind of mutation we have in our recessive disorder or our, monogenic disorder? So one spectrum is called point mutation. A point mutation is mostly the most common cause of the mutation in monogenic disorders. So for example, this is your sequence, TTC. This is your DNA sequence. So when it will be in the messenger RNA, it will be AAG, the opposite one. And then this code, the amino acid lysine. But then if this T is changed, the C is changed to T, so it still encodes the same amino acid, and that is lysine. So this mutation, although there is change in DNA, but there's no change in protein, because we know that one amino acid is coded by at least 2 or 3 codons. So if the codon still encodes the same amino acid, so this change is called the silent mutation. So it's called synonymous mutation or the silent change. Then we have this change of T to C, and that is changing the amino acid. So instead of lysine, now here we have arginine. So this is called point missense mutation. So one is silent, where there is no change in the amino acid, although there is a change at DNA level, but no change at the protein level. So, and we have a lot of silent mutation in our DNA. So from where these mutations are coming from? Anyone?
Genetics, DNA.
हाँ, लेकिन DNA में कहाँ से आती हैं? म्यूटेशन कहाँ से आती हैं?
नाइट्रोजनस बेसिस
जी, नाइट्रोजनस बेसिस तो है ना, लेकिन मतलब वो तो हमारे पूरे DNA में है, ठीक है। अब ये है तो नाइट्रोजनस बेस ही है ना, जो सी जो है वो टी में चेंज हो रहा है या टी सी में चेंज हो रहा है, ये तो चेंज तो आपकी नाइट्रोजनस बेस हो रही है ना, बट ये हो क्यों रही है?
ये कोई एक्सोजेनस इम्पैक्ट्स की वजह से?
बेसिकली आपको जी जी आपको पता है हमारे डीएनए में रेप्लिकेशन होती है, ठीक है। तो रेप्लिकेशन में डीएनए अपनी ही कॉपी बनाता है और उसको जो बनाता है वो पॉलीमरेज है, राइट। सो पॉलीमरेज इज एडिंग, उसके पास एक टेम्पलेट है, ठीक है। आपने फॉर एग्जांपल एक उसको टेम्पलेट दे दिया है कि ये वाली चीजें आपने ऐड करनी है। सो अगर वो while adding those bases to the strand, अगर वो कोई, although our polymerase have this ability and it's a very sensitive process, so nature ने ना बहुत सारे बफर रखे हुए हैं हमारे लिए। अदरवाइज तो फिर एक अगर बेस चेंज होने से डिजीज होती है तो we all should have a disease, but why we don't, most of us don't have diseases or these genetic conditions? Because so once there is a change, अब वो polymerase ने change add कर दिया a wrong nucleotide or a wrong nitrogenous base, but then it will go through again, so it is called proofreading. So पहले उसने add किया and then it will just look like, for example, you write a manuscript and it's exactly writing a book. So you write a manuscript and you put a wrong word, and then you go through that whole thing and you say like, oh no, or maybe you can put some additional word. और अभी तो हमारे वो word में आ जाता है ना, you know, in the proofreading thing that, oh, you have added an extra D here or an extra word here. So similarly, हम एक खुद proofreading करते हैं. So similarly, our polymerase also proofread, and sometimes it's skipped from the proofreading as well. So then we have the DNA when there is a change. So there is a whole mechanism of repair that change in the DNA. So and sometime, and of course that repair, and if it's skipped from that one to pair, that can lead to disease if it's in the coding portion of our DNA. So that, but of course we know that 2% is coding, so rest is non-coding. So that's why if there is a mutation in non-coding, so most of the time it doesn't cause the disease. So these are the point mutations and the missense mutation. Yes, heavy metal effect also, क्योंकि again वो आपके you know they are also kind of something that can lead to the change, and not only the heavy metal but the other germline में तो होती हैं, but of course later even in the somatic mutation, and especially in the case of cancer we have a lot of somatic mutation. So yes, that can be something जो कि आपके polymerase को trigger करता है to make mismatch. So this is also one of the reasons that we have the mutation. And then, so now we have checked that this change that is missense, it's called, now it's changed lysine to arginine. And now if arginine is important, it can cause disease. But if it's not that important in protein, I mean, it's not, iska koi itna heavy function nahi. For example, if you are working in an organization, it's a gene, your department is gene. And for example, if someone have been replaced with someone, another person that comes to replace this person, and it's still doing the same function. So probably there's nothing is gonna affect your department in that way. So similarly, gene में भी सारी की सारी changes disease cause नहीं करती. So, but if the arginine is it comes at a very important position. For example, if it replaces your boss or the head of your department, and arginine is something that is not trained enough or not and doesn't have like, you know, fully training to do that job. So again, it's going to affect in a different way. So that's depend on how important that function of that amino acid is, और कितना उसको ये arginine या जो भी amino acid उससे replace होता है वो कितना उसको कर सकता है. But then there is something जो कि have a huge impact more than any other missense mutation, and that is called nonsense. So one is silent, one is missense, then is nonsense. So nonsense में क्या होता है? If this first T is replaced with A, so it's called UAG or ATC, and that is a stop codon. So there's no amino acid for this one, and its main protein will be terminated. And for example, if you have a protein of 1000 base pair, and this change comes at 100 position, so it's mean you will have a protein of only 100 amino acid, and mostly वो भी nonsense mediated decay में आपकी खत्म हो जाएगी, so you will have no protein basically. So these are the changes. Now I want to ask you, what is SNP or single nucleotide change, SNV or single nucleotide variation, and what is single nucleotide polymorphism? Why all these missense mutations? अब है तो ये भी single एक ही nucleotide change हो रहा है ना, तो single, why all single nucleotide changes are not polymorphism or single nucleotide polymorphism? Yes, we are going to the frame shift mutation later, like in the next slide. So we will discuss that there. So please tell me कि polymorphism, single nucleotide polymorphism और single nucleotide change में क्या फर्क होता है? Anyone? किसी को नहीं आता? चलें हम अभी let's see. So basically जो हमारी ये changes होती है ना, अगर वो बहुत rare हो, if the allelic frequency mean अगर population में आप general population में frequency देखते हैं and वो बहुत rare है, it's mean 0.001% population have it, so it is called rare mutation, or it's called, so single nucleotide mutation is the change that is on one nucleotide, जो कि हमने previous slide में देखा है point mutations. ठीक है? अब वो जो point mutation है, अगर वो बहुत rare है, तो it will cause a disease. But then if it's common in the population, उसकी frequency जो है, वो high है, it's around 1%, so वो polymorphism हो जाता है. So one is very rare, both are single nucleotide change. एक ही nucleotide change हो रहा है. So one is very rare, and that is called rare change, and that can cause all these changes. For example, अभी अगर आप देखें beta thalassemia में ही आप देख लें, so even a small change, one nucleotide change can cause disease, and that is, we know that there's certainly not huge treatment option available for that. But of course, if the change is present in 1% population, it's mean कि वो change इतनी important नहीं है, because वो 1% से ज़्यादा लोगों में है और उन्होंने तो survive कर लिया है ना. So it's if the mutation is rare, it have allelic frequency less than 0.001%, then it's rare mutation, and then if the frequency is more than 1%, then it's a common mutation. So it's kind of a spectrum. और अगर आप इस spectrum को देखें और इस साइड पे आप effect size को देखें, तो हमें पता चलता है कि जो हमारे common variant है ना, जो हमारे जिनकी frequency ज़्यादा है, they have low impact, because if I will sequence my DNA, I might have thousands of common variant. जब मैं उसको उन 6 billion nucleotide के साथ compare करूंगी जो reference genome है, तो I might have a million polymorphism. But why don't I have a genetic condition? Because all those polymorphism are common. So वो इतनी, it's not like that huge impact. And then if there is a change that is not, if that is only in me or maybe like 10,000 other individual in the whole world, and that is causing a rare condition, then I might have a disease. So smaller the allelic frequency, higher the impact, and higher the allelic frequency, smaller the impact. So this is the main concept of this rare and common allele. Tell me if you have any question. आपको ये वाला पॉइंट समझ आ गया? I think so. It's a,
sorry
Ji?
to interrupt. There are some questions in the chat box.
yeah, I saw that there was one related to frame shift, and I just saw that because we are going to talk about frame shift later. But then common variant are good for human. It's a very good question, because now, when we see that why are the most of people in like 70 and 80 and they don't have any disease or the condition. So there are some common variant. There are not all common variant, because common variant can cause diseases as well, and that we are going to talk about the common diseases. But then there are another some variation or some changes, either rare or common, so they can actually protect you from disease. So सारी mutation बुरी नहीं होती. There are some good mutation, and those mutation protect you from genetic condition, and those mutation we call protective variant. ठीक है? तो कुछ ये जो वेरिएंट हमने बात की है ना कि ये कॉमन है, ये रेयर है, इनमें कुछ डिजीज कर रहे हैं, कॉमन वाले कम डिजीज कर रहे हैं, रेयर वाले बहुत डिजीज, I mean they have high impact, they can cause a lethal phenotype even this small change. But then there are some who are protective, who can protect us for genetic diseases, and now we are looking for those one. We are actually sequencing and try to find कि वो कौन से हैं ना, and then based on that we can actually go further for the treatment option और उसकी तरफ जा सकते हैं. So the last line, was it related to this rare and common variant? Someone wrote in the chat the last line. I can explain the rare and common thing, because this is the basic concept जो कि हमें बताता है कि हर change is not disease causing और हर change is not single nucleotide polymorphism. हर SNP जो है वो disease नहीं cause करी, but there are some जो कि common है जो disease cause करी है. So rare is something that is not present in general population. It's a very, very rare, and that's why because of course, because of the rarity, if it's in the important domain of the protein or the important function, it will have a huge impact on the phenotype. But then if it's like common, it's बहुत सारे लोगों में है and they are normal, so of course it is not going to have the same impact. So that's why we call it common variant. But now we know that the common variant also causes diseases, that we will talk later. Acha, now the next is, so here is the picture of them. जितनी rare mutation होगी उतनी, अब वही वाली जो variation हमने इधर देखी है, rare है, फिर low frequency है, जो middle वाली है या intermediate effect है उनका, and then we have a common one. But here, if we look at the spectrum, so if there is a thing that is very, very rare, so it can be lethal. I mean, अगर वो एक single change आ जाती है, so you're and that's very important in development. So fetus survive ही नहीं कर सकता. तो ऐसी condition को हम embryonic lethal कहते हैं. मतलब वो because of the importance of that gene and that protein, if that protein is not present, तो वो for example अगर आपकी कोई protein है जो आपकी जब formation हो रही है, ectoderm, endoderm and mesoderm, and then they are separating, so आपके जो different cells की जो migration होती है कि एक्टोडर्म वाले एक्टोडर्म में जाएं, एंडोडर्म वाले एंडोडर्म में जाएं, मेसोडर्म वाले उधर जाएं, तो वहां पे वो उनका डिस्ट्रीब्यूशन ही नहीं हो पाता, तो then it will not survive. So but then there are severe Mendelian disorder, intellectual disability, epilepsy, there is a huge spectrum of muscular dystrophy and all these conditions. So those are the conditions that are where the fetus is survived, fetus or the child have survived, but of course with the condition just may wo uski it depends, it's not kind of easy, like it needs help or survival ke liye wo, or for example the thalassemia is one of the example as well. So and then we have milder Mendelian condition, for example we have polydactyly, so it's kind of an additional finger, or like if we have some defects in our finger, but of course certainly it's not a lethal condition, so usse koi hamari overall jo health hai us pe koi fark nahi pad raha, you know. So this kind of the thing, so those are the ones jo ke intermediate hai. And then we have ab hum aista aista jo hai na wo common की तरफ जा रहे हैं, ठीक है. पहली वाली बहुत रेयर है, फिर स्फीयर मेंडेलियन है, फिर माइल्ड वाली हैं, एंड देन फिर वो वाली हैं जो कॉम्प्लेक्स वाली हैं, जिसमें कुछ कॉमन वेरिएंट जो है ना, जब वो एक कॉम्बिनेशन में आ जाते हैं तो वो फॉर एग्जांपल अब वो if I have inherited these common mutations, common variant from my parents, and they were predisposed for diabetes, so it's mean like I'm also predisposed for diabetes. So abhi ye hai ki I have the genetic risk of diabetes, but of course it's genetic risk, और इसमें अब मैं environment को अगर change करूँगी तो then I might be kind of will not have the same disease. So this is one thing. And similarly जो common phenotype की variability है, because if one person have 200 common variant for one condition and the other person have 100 variant for this common condition, so might be that condition, the severity of diabetes or like any other condition that is common is variable. For example, in schizophrenia, we know that if the person, there is a huge variability and it might be coming from these SNPs and these common SNPs. So this is the spectrum. तो पहले वो जितना rare होता जाएगा उतना lethal होता जाएगा. So this is the whole thing. So now we come to the point where we have other mutation, like पहले तो हमने एक बड़ी mutation की type की बात की, that was chromosomal, then we actually talk about small chromosomal deletions, then we talk about the point mutations, and now we are talking about the mutation, those are bigger than point mutation but smaller than the chromosomal changes. So these we call indel. So indel can be 2 base pair, can be 20 base pair, can be like it have a specific length, but of course certainly it's not the deletion or insertion of whole gene, it's just the deletion or insertion of a small sequence. So for example, if you look at the wild type sequence, ये आपका जो CATAA, sorry, I have to drink water. Sorry. So then there is a 3 base pair deletion. So the last A is deleted, and otherwise, if you look down in the orange one where we have insertion, so उस sequence जो आपकी wild type थी उसके अंदर 4 base pair insert हो गए हैं. So this is either deletion or insertion is called indel. अब ये indel क्या करती हैं? So indel can be 2 types. One can be frame shift and one can be in frame. So frame shift would be someone that change the open reading frame. ठीक है? और वो ऐसी changes होती हैं जो 3 पे divide नहीं हो सकती. For example, if you have 11 base pair deletion, if you have 5 base pair deletion, if you have 1 base pair deletion, if you have 2 base pair deletion, 4 base pair deletion, 7, तो ये सारी की ऐसी हैं जो कि आपके 3 पे divide नहीं होती. और 3, why the 3 is important here? Because we know that each amino acid is coded by 3 base pair. Like for example, this is ACG and it's called threonine, I think so. And then AGG is arginine. So, but now we have what happened, this one one nucleotide is deleted. So because of this deletion, now the set of 3 amino acid is different. For example, previously we had ACG, now we have CGA, and that is coding another amino acid, that is arginine. So point mutation में तो एक point पे change आने की वजह से वो एक amino acid change होता था. Frame shift में आपका पूरा frame disturb हो गया. So now it's kind of that you have made a different protein altogether, and mostly isme in the frame shift we have early stop codon. So because of course ye wali jo aati rahengi, so that can have jo कि normal protein का stop codon आना होता है उससे पहले stop codon आ जाता है. And these are mostly someone have asked that are the more lethal. Yes, because it depends कि वो protein, आपका whole protein का open reading frame change हो गया है, and then you have a premature stop codon, and that can lead to a very lethal condition. So this is the spectrum of indel if it's not divided by 3. अच्छा, अब if there is an indel that is divided by 3, तो उसका क्या impact होता है? आपको पता है कि 3 का codon है, so अगर आपके 6 base पेयर add हो रहे हैं, तो इट्स मीन आपके 2 अमाइनो एसिड ऐड हो रहे हैं, बट आपकी प्रोटीन के उस पे कोई फर्क नहीं पड़ रहा. The organization of the protein is not disturbed. So it's like you have a piece of puzzle, like you have a puzzle and then you add something into that on the top, but the rest of the puzzle is the same. लेकिन अगर आप उस puzzle को disturb कर दें इस तरह से कि कुछ आप ऐसा निकाल लें कि when you put all these things together, so it make a different kind of a thing. So but then we have in frame. So in frame is either if you delete 3 base pair, so it's mean like you have 1 less amino acid, and if you delete, for example, 12 base pair, so it's mean you have 4 less amino acid, but the rest of the protein is same. So and then the question comes whether these in frame mutation can cause disease or not. So there are some in-frame deletion that cause diseases. For example, अगर आपका वो जो amino acid है वो आपकी ऐसी domain protein में है जो कि important है for the function, so then it will cause disease. For example, this change is not, this addition of couple of amino acid is not that important, so it will not cause disease. So this is the whole thing. Okay, now I would like you to tell me कि ये कौन सी mutation हो सकती हैं. The grey cat run down the hall. अब इसमें पहले एक आ रही है, फिर दूसरी आ रही है, फिर तीसरी. So I want you to tell, unmute yourself, ask me question if you have first, and then tell me if you, what are these mutations. Anyone? So the grey cat run down the hall. It is wild type. So what is the first is point mutation, right? And then we have second one. Third is deletion, definitely. Okay. Third is deletion, second is missense. Yeah, only one base pair or only one alphabet is changed. Yes. What is the last one? Last one, yes, last one is nonsense because उसपे वो जो भी mutation आई है उसकी वजह से protein मतलब नहीं बनी ना, so the gray doesn't make any sense. So now you can actually get an idea that what these mutation and what these variant can be or which kind of effect. For example, अगर nonsense आ जाती है तो वो तो सारा के सारा उसने protein मतलब उसे the gray, does that make any sense? ठीक है, तो ये वाली जो है, these are the things. Okay, I think so we are done with this part, but I would like you to ask me questions, and then of course we can we have a 30-minute break if you want to take the break. I don't know if we have to go with the thing, or we can start and we can utilize this time in our next lecture. Structure defect or enzyme defect more commonly. Structure defect or enzyme okay, both. Commonly depend करता है कि if it's the gene that is in the structure जो है reference important है, and if it's in the gene, because now we know that we have a lot of conditions जो कि metabolic disorders हैं, ठीक है, and those are all the enzymes and those things. So depend करता है कि किस gene में है. I don't think so there is, when mutation happen तो उसका कोई as such नहीं होता कि you know it is going to be in enzyme वाला gene or it is going to be in the structural gene. So I don't think so there is any relation that is common between structural and enzymatic, but we know that there's a lot of enzymatic conditions. So any other question? Okay, okay, then let me ask you a question.
Excuse me, we can continue to 11. So 11 to 11:15 we'll take a break. Then we'll come back at 11:15. अभी आप 11 बजे तक पढ़ा लें.
अच्छा ठीक है, तो मैं फिर अपना अगला लेक्चर शुरू कर लूं?
जी जी कर दें. 11 to 11:15 we'll take a break, short break.
11 to 11.